Sorry about that. I think I'm ready now. Zoom in. There we go. Okay, so yes, Chapter 6. Finishing Chapter 6, looking at enzymes, which is all about like metabolism and how chemical reactions occur. So 2 sections here. Whoa, what the heck? Golly. There we go. So enzymes, they are biological catalysts. That means they help start chemical reactions by lowering that activation energy. The substances that interact with enzymes, we call them substrates. So in our liver lab, H2O was a substrate. It binded to catalase, that was the enzyme, and then the enzyme broke hydrogen peroxide down into water and oxygen. So substrates, the molecules that undergo a reaction, bind to the enzymes. An enzyme is a unique protein 3D shape that stabilizes a temporary association. Things that enzymes do, they lower that activation required for new bonds to form. So as a result, the reaction proceeds more quickly with the enzyme versus without. The enzyme is not accounted for in the chemical equation, and that's because it's not changed, it's not used up, it's reusable. You only need a small amount of it. So I want to talk a little bit about the active sites of those enzymes. So the active sites is where the substrates actually bind to. And a lot of times active sites, you see like a groove, a pocket, or a cleft. Maybe like you can kind of think of it as a lock and key model where the lock is the enzyme and the keyhole, like that the key fits into, would be the substrates. And so enzymes tend to be very specific. When that substrate binds to the enzyme, you call it the enzyme substrate complex. And like I said, the substrate must fit precisely. It's very, very specific to that enzyme. Chemical reaction will occur. Maybe bonds will break, maybe bonds will form, but eventually you're going to get a new product or products. And then once that is formed, it will dissociate or fall away from the enzyme, and the enzyme will go and find a new substrate to bind to and repeat this cycle. So this diagram shows this process where you have your enzyme and the grooves that are very specific. We call that the active site. That's where the substrate binds to. This is the substrate. And as you can see, this nice little rounded shape with the triangular, well, it's going to fit nicely into this substrate. So the substrate enters the active site of the enzyme. It forms the enzyme substrate complex. Okay, now a chemical reaction will occur. In this case, it looks like it's going to break the bonds. Okay, and now you get 2 new products, and this enzyme will go around and find a new substrate to bind to and repeat the process. Another diagram showing the substrate bind into the active site, you get your enzyme substrate complex. Like I said, enzymes, very specific, so only certain substrates will fit into it. The rectangle will not, bind to this enzyme, and the chemical reaction will not occur between those 2. So they are very specific to what they combine to. So there are many forms of enzymes because enzymes are proteins, and proteins, wide diversity of them. Some enzymes kind of line up in an assembly line, and when that happens, you call it a multi-enzyme complex. So you just have like one enzyme after another that carries out a huge process. So in cellular respiration, we're going to talk about pyruvate. Pyruvate comes from glucose. Basically, glucose, does anyone remember how many carbons are in glucose? 6. Very good. Okay, so this is glucose. And then what happens in the process of with cellular respiration is glucose gets split into pyruvate. And pyruvate is a 3-carbon chain. Why do I hear footsteps? Thank you. Okay.
Just trying to sneak in and just leave it for you. Not interrupt you.
I was like, what is that? Okay, so at the end of glycolysis, you end up with a 3-carbon molecule called pyruvate. And this happens in the cytoplasm of the cell. But then pyruvate latches on to a coenzyme, and it gets transported into the mitochondria, where it goes through a series of reactions, all carried out by different enzymes. And so for when we get to cellular respiration, you won't have to memorize the enzymes, but you do have to memorize the intermediate molecules that are formed after each enzyme. And here's a hint to help you study for it. When we get there, a lot of times the enzyme is named for the molecule that it makes. So then on your diagram, all you have to do is look at the previous enzyme, and it might help you, oh yeah, that's where 1,6-bifructose goes, or whatever intermediate molecule is made. These enzymes that are in the multi-enzyme complex, they have to be in close proximity to one another because it's literally an assembly line. It takes it and modifies it, moves it, passes it on to the next enzyme, modifies, breaks it down, cleaves off things, so on and so forth. So one example of a multi-enzyme complex is the pyruvate dehydrogenase complex, where you actually have three enzymes lined up, and it looks like pyruvate comes in, we cleave off a carbon, so now we get a 2-carbon molecule, so it's called pyruvate dehydrogenase 1, and then I can't even read that, oligohydrofructidylase, I don't know, but whatever. These enzymes lined up, and eventually they make a new intermediate molecule, in this case acetyl-CoA. So there are some advantages with our multi-enzyme complex. So the rate of the reaction is limited to the frequency of collisions. So in order for a chemical reaction to occur, they have to meet. Well, if you have an assembly line, they're going to be in close contact, so it's going to carry out this reaction a lot faster instead of having those molecules diffuse away and trying to find the enzymes. Reactant substrate never leaves the complex during the reactions, so it reduces any unwanted side reactions that could occur. And then finally, all reactions that take place can be controlled as a unit. So we're going to talk a little bit about how chemical reactions are controlled by inhibitors and activators. I do want to stress, though, that not all enzymes are proteins. There are some non-proteins, and we call those either coenzymes or a cofactor. So just understand that, enzymes are usually proteins, but there are some that are not. So like I just mentioned acetyl-CoA, well, that would be a cofactor or a vitamin of some sort. So a lot of vitamins that we consume, they're actually coenzymes, and minerals are cofactors. Remember, with enzymes, the environment that it is in does affect its function, and we did look at this with our liver lab. You can change the temperature, and a lot of times if you increase the temperature of an uncatalyzed reaction, the reaction will increase to a certain point. There's usually an optimal temperature before it starts to teeter off and the reaction doesn't work anymore because you've unfolded the proteins. So increasing the temperature in an enzyme catalyzed reaction will increase until that certain break-in point, if you will. If you're below the optimal temperature, the enzyme shape is not as flexible, so it might carry out the reaction but at a slow rate. If you're too high above the optimal temperature, it's too weak, the enzyme shape denatures, it unfolds, and now it's not really going to work well anymore. And then what's the other way that you can change the enzyme function? So temperature and pH. Thank you. So pH, changing the hydrogen ion concentration. So a lot of times, what's holding enzymes together are those ionic interactions that we talked about with protein level structure. And these interactions are sensitive to that hydrogen ion concentration. Most enzymes have the optimum pH that ranges from 6 to 8 because in living organisms, that's usually the pH of what's inside them. Because all living things, or I should say cells, are mostly just made up of water, and water is neutral, but they can be slightly acidic or slightly basic. So here, this graph is looking at different enzymes that are inside of us with enzyme activity, and then the pH values, 7 being neutral, acidic, basic. So salivary amylase tends to be, very optimal at pH 7. And if you just think about what's in your saliva, it's pretty much water. Pepsin, which breaks peptide bonds in the stomach, has an optimal pH of 2. And that makes sense because hydrochloric acid is also found in the stomach. And then arginase which I don't know where that is at. Probably should look it up. My guess would be somewhere maybe in the small intestine, so past the stomach. A little bit more basic here with pH value of 10. Okay, so controlling chemical reaction rates with enzymes, we do have some inhibitors and activators. And so when these inhibitors or activators bind, they're either going to stop the process or they're going to expedite or increase the efficiency of it. And so with this inhibition here, here's my enzyme. There's an active site for the substrate. It binds. You get that complex. You make the products. But if you get an inhibitor, notice that the inhibitor, in this case, has the same shape, but it's got a little bit different top to it. Well, this inhibitor will bind to the enzyme, and it will continue to stay bound to that enzyme so that the substrate can't bind to it. So it's like out-competing it, so this chemical reaction will not take place. So this is one way that you can regulate it. So there are different forms of inhibition as well as activation.
So the pH will either stop the reaction or speed it up.
The pH,
The inhibitor.
the inhibitor, yeah, or activate. So inhibitor means that it will slow it down, okay, or stop it. And activate means speed it up or give it a green light to carry out the reaction. So an inhibitor is a substance that binds to the enzyme and decreases its activity. A lot of times in a biochemical pathway, it kind of like self-regulates this process in a negative feedback inhibition. But inhibition can occur in two ways. You can be competitive, so it will compete with the substrates for the same active site, or the non-competitive inhibitor, I'm going to call it the sneaky inhibitor, and I'll explain why, but it binds to a site other than the active site on the enzyme, and as a result causes the enzyme to change shape so that substrate doesn't fit nicely into that active site anymore. So I have diagrams that show competitive inhibitors and non-competitive inhibitors. So with the non-competitive inhibitor, okay, let's see here. I'm just trying to figure out. So this is the substrate. It's supposed to fit in here, okay? But the inhibitor bound, is that the word I want? Binds to a site on the enzyme other than the active site, causing a configuration change. So instead of this being open, it closes, and the substrate can't fit in there nicely anymore. Lock and key. So that's one way to shut it down. So this is non-competitive because it's not competing for the same site. But the competitive, okay, it binds to the active site that the substrate wants to bind to. So it kind of like beats it there, kind of like musical chairs maybe. But the substrate, oh shoot, someone's already there. Okay. So then the reaction never takes place. Enzymes can exist in either active or inactive states, so we call it allosteric enzymes. And this is a term we have gone over previously, like allosteric molecules. You know, like they have the same chemical formula, but, you know, it might be rearranged a little bit differently, or left-handed versus right-handedness. Same thing with enzymes. So these allosteric sites place where the non-competitive inhibitor binds, and it tends to serve as an on and off switch. If the inhibitor is bound to the enzyme, it's off. If the inhibitor is not bound, it's on. So you can have these allosteric inhibitors that bind to that site, or an activator which can actually turn on the process. So here's an allosteric inhibition here. My enzyme, the active site that the substrate will bind to, here's my inhibitor. It binds to a different site, that's called the allosteric site, and as a result, instead of being moon-shaped, kind of just like closes its little mouth, it's not a mouth, but it looks like a mouth, and now the substrate doesn't fit nicely anymore, so we've shut off this enzyme. With the allosteric activation, it means we're going to turn it on. So this enzyme is off, moves the little mouth, here comes the activator, induces a change in the enzyme shape. Now it's moon-shaped, the substrate binds to it, reaction is carried off. So inhibitor decreases activity, and an activator increases it. So again, did I just go backwards? What is up with my PowerPoint? It went back 3 slides. I was like, I've already said this. Okay. Last, well, it's not the last, actually. Yes, okay. Last thing about enzymes, so we've talked about inhibitors and activators. We also have those enzyme cofactors, so they're not proteins. They're just additional components that assist an enzyme function. A lot of times with cofactors, it's a metal ion, and they take electrons away to make the bonds easier to break. So remember, we talked about how you make a bond. You can share electrons or you can transfer. Well, these guys kind of steal those electrons so the bond happens more readily. If it is a non-protein organic molecule, we call it a coenzyme. Remember, with the transfer of electrons, redox reactions are taking place. And so in this case, our coenzyme is the electron acceptor in this. So if you accept an electron, is it being reduced or is it being oxidized? Think about the saying oil rig. Say that what? So it accepts the electron, so it gains. So it's actually being reduced. It's opposite of what you think. Right. So if you get, yep. So oil rig, oxidation in loss. Reduction means, reduction in gain of electrons. So they gained electrons, the coenzyme's being reduced. So here's an example of a cofactor working with an enzyme. So we have this active site, but it's incomplete. The substrate, parts of it match up, but not all of it. So this cofactor will come in with the reactant substance, and now it fits perfectly, and the reaction will take place. So that's how coenzymes work. Okay, I think this is the last section, and it's titled Metabolism. Basically, metabolism is any chemical reaction that's carried out by an organism, but there's actually two types of metabolism. You can have animalism or catabolism. And with animalism, okay, you tend to make large molecules. You're forming bonds, but you need energy to do that. So it will expend or use energy to make or transform bonds. So you have all these little small molecules. With energy, we can form bonds between them and make a very large molecule. So that's animalism. Catabolism means you're going to harvest energy and break bonds. So I have my large molecule, I'm going to break it down into small molecules and use that energy to carry out other chemical reactions in the living organism. So catabolism, break it down, anabolism, build it up.
Is it cannibalism?
Yeah, that's how I remember it.
Yeah.
The regulation often depends on this feedback mechanism, so I'm going to talk a little bit about that here. So the end product in a chemical reaction might bind to an allosteric site in an enzyme and catalyze the first reactions. So there's this feedback inhibition where the products can actually go back to the beginning and control how fast or how slow the reaction is. So in my example here where C gets modified to D, D gets modified to E to F, and then if I combine it with a G, I get H. Okay, so this is just like a simple example. Okay, so as H is increased, it might encounter an allosteric site on the C to D process that would bind to the enzyme responsible for turning C into D and shut it off. So that's one way that things can be regulated. So I guess I should, when I talk about this diagram, these are like the products, and the arrows are the enzymes responsible for turning the previous product into the new product. Oh, yeah, there's my arrow. Yeah. And then it stops that process. I forgot I had little animations. Okay, wow. That's it. I thought I had one more slide. Okay, questions about inhibition. Basically, it's with the biological mechanism, the product of it will somehow go back and shut off something before it, so no new H's are made. Okay, questions about enzymes? No? Any questions from previous homework that you might be struggling with? well, then we'll say the Chapter 6 guided notes are due tomorrow. And then, yeah, prepare yourself for lab. Please do yourselves a favor and just read through the lab so you get an idea of what you're trying to do. So Herman Norcross, you have distilled water, and we'll take care of the sucrose concentrations. And I'll make that Google Docs and try to share it with everybody. I'm hoping because we have different domains, we can still share things. I think so, because James shared a document with me, so we should be okay there. All right, so I'm just going to sit here, and if you have questions, just holler.